In this video, we will discuss about the T cell glycosylation and how this glycosylation on the T cells will lead to pro-tumorigenic or anti-tumorigenic response. So this in the middle of the left hand side figure, it shows the type of glycosylation that are found on the T cell surface, and we can see that most of them are complex and linked glycan. However, some of them are also O-linked glycan that can be seen here. So uh, now let us these glycans helps in the interaction with the ligands or this which are commonly known as glycan binding proteins, and that will leads to the several T cell function. For example, this galactin bindings such as the galactin 9, the galactin family binding proteins such as this galactin 9, galactin 1, galactin 3, or this beta galactosidase will cause will helps in this T cell receptor activation, or we can say it determines the threshold above which T cells will get activated. The next type of glycans are the silated glycans such as the Siglac9, and this type of glycans helps in the inhibition of the immune response. So this types of glycans prevents the autoimmune response, we can say that. The next type of glycans is this silofucosylated glycans. You can see the fucose structures on the glycans below it, and this some of the example of these silofucosylated glycans are the L-selectin, and this helps in the migration of the T cells to the site of its requirement, or we can say T cells migration, maturation, or development, that we have discussed in one of our previous videos, that how these T cells migrated from bone marrow to the thymus for its development, that is it converted itself from double negative to double positive. And now what let us understand what will happen if these glycan structures got disrupted, and the uh, this will lead to the weakened interaction of this glycan with this glycan binding proteins and of the T cells. So if the glycans that plays an important role in galactin bindings get disrupted, then it will cause this increased activation or reduced apoptosis. So this might lead to the case where it will generate a pro-tumorigenic response, as it will the threshold levels for this activation of TCR will increase, and also the reduced apoptosis will also increase, so that it will trigger less, since the threshold level of TCR is increased. So its functioning to kill the uh, bad cells or the tumor cells will get reduced. The next type of glycans is the silated glycans. The uh, um improper functioning of the interaction of the silated glycans with the T cells will lead to the increased activation or the autoimmune response, as the silated glycans helps in the inhibition of the immune response, and this will cause a reduced one. This will also impact the this galactin 1 binding and apoptosis. And the next kind of the last category of the glycans that get uh, that affect these glycan binding proteins are the silofucosylated glycans, and it will lead to the increased rolling, or we can say that T cells will not be able to reach to a place where it is required, and the extra vascularization, maturation, and activation will increase, so that the T cells might divert it to the wrong pathways, we can say that in general term. And there is a table that determines, or that whatever we have discussed in the figure has been tabulated in this table. Let us just quickly go through in brief that these galactins kind of functions. It will cause this galactin 1 the improper interaction with the galactin 1 will cause a pro-tumorigenic effect, whereas galactin 3 will cause this anti-tumorigenic effect, because the improper functioning of galactin 3 will prevent the T cell apoptosis, thus it will cause anti-tumorigenic effect. So that could be one of the therapeutic targets that can be used against the tumor cells. And this extracellular negative regulation, this galectin 3 which also has some pro-tumorigenic effect also. That is, it will cause negative regulation of T cells by destabilizing the immunogenicness or promoting down regulation of T cell receptors. Since the T cell receptors get down regulated, it will not be able to perform its function regularly. So the T cell receptors such as these CTLA4 and LAG3 will be reduced in the case of this galectin 3 improper functioning, galectin 3 improper interaction with the T cells. So these are known as checkpoint inhibitions. Checkpoint inhibition. So these are not reduced. This will be overexpressed on the T cells when this galectin 3 functioning is not interacting with the T cells. So overexpression of this CTLA4 and LAG3 will lead to the inhibition of these T cells or increase in the threshold of the T cell receptors. Thus, it will cause pro-tumorigenic effect. This galectin 9 will negatively regulate T cell immunity by binding to the TIM3. So thereby uh, it will also increase the threshold of the T cell receptors and thus helps in the tumor for its uncontrolled growth rate. And this in terms of selecting or glycosylated glycans on the T cells, uh, this its acquisition on the T cells will increase its T cell effector function. That will be helpful uh, to target the T cells. On the other hand, these L-selectins on the leukemia cells will cause improved trafficking and distribution abilities, or we can say that it helps in the metastasis. Thus, it it comes on the pro-tumorigenic effect. And in terms of SIGLEC, that is on silated glycans on the TIL, it will affect the memory T cells and with higher functional responses after stimulation. So in other terms, it will increase the immune suppression on the T cells because of this SIGLEC9 inhibitory function. So thereby it oh, this will not suppress the immune response. This, we can say, this will overexpress the immune response. So the function of these silated glycans is to suppress the uh, T cell response. However, the we can say the less expression of the silated glycans will lead to the high functional response after stimulation. So it will be helpful to target and kill the tumor cells. And the select 19 on tumor cells will cause the suppression of these T cell responses. Thus, this will promote the tumor growth within the host. So these are some of the overviews how this glycosylation on the T cells function, and how this uh, improper glycosylation on the T cells will cause disruption in the immune response, and how they are related to pro-tumorigenic or anti-tumorigenic responses. If you need to understand more about this role of glycans on the T cells, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.